So thank you for being here to this Journal Club of May, exceptionally on a Tuesday, and not the first Tuesday of the month either. Today we are assisting and commenting a presentation by Kanchan Kulkarni, who's a postdoc by most of you know her. She's a postdoc. I don't know if are you still a postdoc?
I'm a researcher now, but
researcher, great. Congratulations. At Lyric, working with Olivier and Rick, especially on the field of cardiac alternance. So this changes in the morphology and properties of parts of cardiac signals. In this case, we will see the ECG, clinical ECG, and how this relates to cardiovascular diseases, in particular with arrhythmias. So Kanchan will talk to us about P-wave alternance, which is, let's say, a different approach for people who might be used to T-wave alternance in the ventricular context, and something that we are very interested in. So I'll leave you the stage, Kanchan, for your presentation. Thank you.
Thank you, Guido. I'll share my slides. I don't know how to change it out of the presentation mode.
Well, okay.
But okay. So as Guido mentioned, this topic, this paper covers P-wave alternance, which is not as commonly studied as ventricular alternance. But the timing of this journal club was great because this paper is literally hot off the press. It was just published 3 weeks ago. So hopefully everyone will find it interesting. I'll just start with some background. So this paper is published by a group called Richard Verrier and Bruce Nearing. They're a highly renowned and established group from Harvard Medical School and the BIDMC, so Beth Israel Deaconess Medical Center in Boston. They've been publishing about alternance from the early 90s, so essentially since I was in diapers. So they've been doing this work for many, many years. And this paper, as I said, focuses on atrial arrhythmias, atrial fibrillation. But before we dive into that, I thought I'll just cover the topic or the background of alternance and techniques to measure alternance using ECG in just a couple of slides. Background, a lot of our knowledge about alternance is related to ventricular arrhythmias and ventricular alternance. And it's been suggested and proven using few studies that before the heart transitions from normal sinus rhythm into tachyarrhythmias, chaotic, unsynchronized electrical activity, it transitions through a specific rhythm called alternance, which is a beat-to-beat alternation in the action potential duration or amplitude. So on the ECG, it manifests as this alternation in T-wave morphology or duration. Usually it's very subtle, so it's in microvolts, so it may not be as clearly visible as this image shows. So you need advanced signal processing methods to really extract it and quantify this from electrocardiograms. At the cellular level, it's more apparent. It's this long, short alternating pattern in the cellular voltage.
I think it's essential that the cycle length remains stable.
Yes.
Because it can be secondary to changes in the cycle length, B2B changes in RR interval, for instance.
Absolutely, yes.
And I think the prerequisite for alternances is that the RR interval is the same.
Absolutely. So yes, that's a major factor when you're trying to use these signal processing methods to extract alternance, that the underlying heart rate has to be consistent and constant during the recording.
Yes. Yeah.
And this phenomena has been associated to a number of pathologies. It's a substrate-dependent phenomena, so it's suggested that alternance depicts sort of substrate properties. It's been associated to infarction, heart failure, different pathologies, but we won't go into that. Instead, we'll come to atrial alternance. So just as T-wave alternance have been linked to ventricular tachyarrhythmias, in the more recent years, people have started to look at atrial alternance, so specifically alternation in P-wave morphology or duration, and try to understand if it is similarly linked to atrial tachyarrhythmia, so atrial fibrillation. there's been studies that have looked at cellular atrial alternance, and they have shown similarities in terms of mechanisms to ventricular alternance. So, for instance, these recordings taken from atrial myocytes in a paper by Kanopoulos and Blatter back in 2017 showed similar calcium transient fluctuations in atrial myocytes that lead to action potential duration alternance, as you see in ventricular myocytes. But there are important mechanistic differences in atrial and ventricular alternance because atrial structure is different, its ionic mechanisms are different. But an important factor that will come again in this paper for us is that we're looking at P wave alternance, and P waves denote depolarization, as opposed to T waves denoting repolarization. So the underlying fundamental mechanism that people are investigating here is different. When we are looking at P wave alternance, we are looking at abnormalities stemming from probably sodium channel abnormalities, versus when we look at ventricular or T wave alternance, we're looking mostly at calcium cycling and those kind of repolarization abnormalities.
Can I ask a question about that?
Sure.
Sorry.
Because I think the P wave, the action potential in the atrium is more or less triangular, like you show here, and therefore repolarization is almost, connected to the start of the activity. So I think you can't state that the P wave is only related to depolarization. It may as well incorporate changes in the repolarization, I think.
Possibly, yes. Most of the studies say that, yes, that there might be a portion of repolarization included. But I think in terms of saying that a lot of the repolarization of the atria actually go into the PR interval and also get overlapped by the QRS complex sometimes. So it's difficult to identify the complete repolarization. But at least you get a sense of the depolarization just by looking at the P waves.
Okay.
Can I also ask a question on this? So typically to get the alternans, you have to accelerate basically the cycle length, right? So you want to shorten that to accelerate the rate, which will usually exacerbate, for example, T wave alternans. But however, doing that, wouldn't you suspect the T wave and the P wave to start merging with each other? At what rates do you study the P waves? Like what pacing rates?
So again, it depends on the methodology that you use to extract alternans. So when you're using the spectral method, a baseline rate of 100 to 110 beats per minute is recommended. So at that rate, you still pretty much have very consistent P and T waves that don't overlap. When you're using the modified moving average, which is actually used in this paper, you actually are not restricted by a baseline heart rate, which is why sometimes this method, the time domain method, is preferred, because you can use it in different pathologies at different heart rates in different conditions. But it also has its trade-offs, which we can, I guess, discuss once we go into the method a bit more in detail.
Thank you.
One more question, maybe. Just
Yeah.
Chantal from Amsterdam. A different P-wave morphology could also be caused by, for instance, areas of block, and 2-to-1 block in the Bachmann's bundle, for instance. Do you think that could play a role here, or do you think it's really an electrical phenomenon on cellular level?
So I'm not very familiar with the alterations that occur in the morphology due to the block. But in case of alternance, we are looking for a very specific change in the even and odd alternating beats. So we can look at it when we discuss the methods. So we're looking at a very specific pattern with a specific frequency that should only occur with this underlying phenomena.
Okay, thanks.
So as we just mentioned, there are different methods of extracting alternans from the ECG: spectral method, moving average method, more complicated nonlinear methods. But only two methods have been clinically approved, which is the spectral method that does a fast Fourier transform, and the modified moving average method, which is actually used in this paper. The spectral method, as we talked about, the main sort of caveat or disadvantage is that it requires a steady underlying heart rate of 100 to 110 beats per minute. But what it does is this essentially takes a sequence of consecutive beats and it plots a power spectrum. and tries to quantify the power in the alternance frequency. So the alternance frequency is 0.5 cycles per minute. It's, as the definition says, it's an alternating pattern, so it occurs once every 2 beats, so 0.5. Won't go too much into the details of this, but that's the basic phenomena of that method. The modified moving average, which is more relevant to this paper, is a completely time domain based method. So in this, say if you have a sequence of beats, as shown here, 6 beats, you divide the beats into even and odd beats. So in this case, the first, third, and fifth beat are classified as group A, the odd beats, and second, fourth, and sixth beats are classified as group B, which are the even beats. Then you essentially just take an average of group A, an average of group B, superimpose the 2 beats, and find the difference between the maximum, find the maximum difference between the 2 beats. So that gives you a measure in microvolts of the level of alternance, if that makes sense. So with that sort of basic knowledge, we go into this paper. So in this paper, they're focusing on patients with atrial fibrillation, which is one of the most prevalent sustained arrhythmias in adults, affecting 40 million people worldwide. And it also significantly increases risk of mortality. And the most common treatment strategy is pulmonary vein isolation. So ablating the ectopies that originate from the pulmonary veins. And the paper also states a couple of other studies that try to relate alternance and atrial fibrillation. Again, there's not a lot of work that has been done in atrial alternance. So there are only these few studies that showed that action potential duration alternance can precede the onset of atrial fibrillation, and that this action potential duration alternance are more sizable in persistent AF patients than in paroxysmal AF. And our own study was also cited, which showed that P-wave alternance correlated with increased atrial fibrillation in patients with paroxysmal AF. So this specific paper, the question they were asking is, can a rebound in P-wave alternance level predict atrial fibrillation recurrence after ablation? And the hypothesis, as we very briefly discussed, is that they suggest that increased P-wave alternance level correlate to an increase in action potential amplitude alternance. So it comes down again to sodium channel inactivation dysfunctionalities, which are getting represented in these P-wave alternance. So just to summarize the methods, the patient population consisted of both paroxysmal and persistent AF patients that were admitted at the Beth Israel Deaconess Medical Center in Boston. They were all first-time pulmonary vein isolation patients that were enrolled during a 5-year period, 2016 to 2021. 12 lead ECGs were recorded in 45 subjects, both pre and 24 hours, up to 24 hours post-ablation. And 4 to 16 weeks of follow-up ECG was done in only 30 patients. Arrhythmias were defined as atrial flutter, fibrillation, or tachycardia episodes that lasted greater than 30 seconds. And recurrence was defined as any occurrence of atrial arrhythmia during the first 90 days after ablation. The average follow-up period was 955 days, so it's about 3 years. And they also importantly had 20 control subjects, so ECG recordings from healthy subjects that had no cardiovascular disease or AF. And in terms of analysis, they did P, T, and R wave alternance pre and post ablation and during the follow-up period. And then they did quite a few statistics to look at the survival and sensitivity and specificity of P-wave alternance. So in terms of the method for extracting alternance, it's very similar to the modified moving average we discussed. The only difference being they actually applied artificial intelligence to improve the beat separation. So they applied a convolutional neural network-based machine learning model to the raw ECG that helped them to remove artifacts and better identify true segments of alternance. So to better separate the even and odd beats. Unfortunately, a lot of the details of the artificial intelligence part of the algorithm were not described in this paper, but they published a separate paper just on the technique back in December 2023 that describes this process, if people are interested in following up. But after the AI part, the rest of the algorithm was the same. the beats were sorted into even and odd beats, averaged, superimposed, and then the difference between the P waves gave us the alternance value in microvolts. So in terms of results, there were 45 adult patients. Most of them were paroxysmal AF, so about 86%. The rest were persistent. And this is the main sort of crux of the paper, where they saw that P wave alternance was reduced significantly by 48 to 63% post pulmonary vein isolation. And they found this phenomena in only 3 leads, lead 2, lead 3, and lead AVF. Pulmonary vein isolation also reduced R waves, so QRS complex alternance, and T wave alternance by 49 and 47%, which is interesting. And they showed that at follow-up, so during the 4 to 17 weeks after ablation, P wave alternance rebounded by 125% in the patients that demonstrated atrial arrhythmias. So this was their main finding, that at follow-up, patients that showed an increase in P wave alternance demonstrated recurrence of atrial arrhythmias. And they found that this threshold of 4 microvolts was really efficient in identifying patients that had recurrence of atrial arrhythmias. It had a sensitivity of 88% and a specificity of 82%, which is quite high. And looking at the survival characteristics, so they found that the first average time to the first arrhythmia was around 604 days. The hazard ratio for a threshold of 4 microvolts was 3.4. So basically, patients that had P wave alternance values of greater than 4 microvolts at the first initial follow-up were more likely to have atrial arrhythmias. So recurrence of atrial fibrillation.
So can I ask a question about this? But follow up on this, is this people that have had, hold on. Gotcha, never mind. I see what, I missed a second there. Thanks. Sorry, no question.
So that's basically the summary of the results. There were 4 figures. So the major findings was that P wave alternance is significantly reduced post pulmonary vein isolation. So this ablation technique modulates the levels of alternance. This is a finding that is new and not shown in other studies. In patients whose P wave alternance remained low at follow-up, AF was significantly less likely to occur. And vice versa, patients that had rebound in P wave alternance later experienced recurrent atrial arrhythmias. So a P wave alternance threshold of 4 microvolts at initial follow-up had a hazard ratio of 3.4 and was very efficient in distinguishing patients that were likely to have recurrent arrhythmias. So they established that P wave alternance may identify a risk of atrial fibrillation recurrence post-ablation. Some strengths of the studies were, as I said, this is the first study to actually evaluate the effect of ablation, pulmonary vein isolation on alternance. They compared pre and post-ablation levels to healthy controls. So they actually showed that pulmonary vein isolation reduced alternance levels to those that were seen in control subjects, which is also an interesting finding. And they were able to perform long-term follow-up of these patients and show the utility of P wave alternance as a biomarker of atrial fibrillation recurrence. But some limitations were it's a very small sample size, so 30 patients, out of which only 18 patients showed recurrent arrhythmias. So it's quite a small sample size to test the utility of alternance. The other thing that I found interesting and not a lot of discussion was that they didn't explain the effect of the lead position on sensitivity and specificity. So why only these 3 leads were able to identify alternance? Is there a dependency of the technique based on where you measure or record alternance from? Also, I haven't read the other paper in detail, but I wasn't very sure what was the need for including artificial intelligence in the modified moving average algorithm, because they don't talk about how the results compare to applying the traditional method, which is clinically approved. So if somebody wanted to apply a clinically approved method to analyze this data, what would that change the efficacy? And also, we don't get a lot of mechanistic or pathophysiological insights from these findings. So yes, alternance is modulated and it might be a biomarker, but how does that actually compare to cellular mechanics? So apart from the fact that maybe sodium channels are involved, we don't get a lot of other insights into this. And it's, you know.
Well done. Good presentation. Any comments?
I just had a question. Do you think it's necessary to know the mechanism of the P-wave alternans in this case if they're showing that they just predict the reoccurrence over a certain time?
So
Yeah.
I think it's interesting because, again, one aspect that the study didn't really differentiate was paroxysmal versus persistent. So if there are different mechanisms that occur or happen in these substrates, then we might have different utility of this technique. Because in our study, we had found alternans to be more, well, easy to extract and also estimate in paroxysmal AF patients. So it's just something that if you know what is causing the alternans, you might be able to better apply it to screen patients.
I agree that for our labs, I think getting to know the mechanism is the challenge. And speaking about that, I think the involvement of the sodium channel is not very likely, I would say, because the diastolic interval of the atrium is much larger than that of the ventricles. So I don't see how the sodium channel can be involved, to be honest. It's very unlike what is happening in alternance during ventricular arrhythmias, or during sinus rhythm in patients with ventricular arrhythmias, where the diastolic interval is almost 0 in order to get alternance. So I think there must be another explanation for it. Do you have any idea of alternative explanations?
Not at the moment. I mean, I studied ventricular cells more than atrial cells, and whenever we see amplitude alternance, it's always accompanied with action potential duration alternance. So we see both APD and AP, action potential amplitude alternance, together. So to just see or think of sodium channel abnormalities leading to this significant level of alternance is, there's something missing there. So I feel like there are other things going on there. There are probably calcium cycling abnormalities which are not being captured by the P waves that are also leading to this abnormality. But it's a question of how you're going to measure them from the ECG, because you can't really measure the repolarization phase from the ECG directly.
Mm-hmm.
It's also a bit strange that it is also affecting QRS alternance and also T-wave alternance. So it makes you think, is there something else going on which is resulting in all these? And also, it's already half a day or the next day after the procedure, I guess, that they make the ECGs. What cellular mechanisms can be already altered in such a short period of time?
Yes, that was actually, very interesting that they saw such a big change, almost like 50% drop in QRS and T-wave alternance, which is,
I found it a bit weird that in such a large center, in 5 years, they can only include 45 patients.
Yes. I think this was more like, it was a retrospective thing, so they probably did the study to measure something else, and then they did this analysis as an add-on.
I guess so, yeah.
I would also like to comment a little bit on this paper. First of all, Kanchan, thanks a lot for presenting it. I really liked how you nicely lined all the aspects of the study. So I will start with a little bit mentioning my background. So I did my PhD in Uli Schutten's lab, which is specialized on atrial fibrillation. And when I was there, I remember there was one project that particularly focused on P-wave alternance and how this can be used as a predictor of atrial fibrillation. And although I don't remember exactly how the study ended up, I remember that there were quite some technical challenges when doing this. First of all, what I remember is actually just the signal processing aspect, which is challenging, because P waves are relatively small and can be very small in some leads, which might explain why they chose only certain leads for the whole study. That's one aspect. Then it's removing the noise and removing the artifacts, which also can heavily affect the whole analysis, because the artifacts can sometimes be as large as the whole P waves, right? Or the noise can have an amplitude of the same amplitude as the whole P wave. So then signal filtering really plays an important role. I also remember they were really like struggling, or there was like some technical aspect of like how to like really like delineate like where does the P wave start and end, because if that's not done correctly, then the whole subsequent analysis doesn't work at all. So I can imagine like all these aspects could be dealt with to some extent with that AI method the authors used. Although at the same time, we might a little bit question that and say, okay, maybe they wanted to make things a little bit too perfect, and some parts of the alternance or some features of the alternating alternance, beat-to-beat alternance, could be just erased by the AI method. So that's more like a comment. Another comment comes just to like a very simple thing is like signal processing, that some filters, since this is I guess digitally recorded ECG, not always the filters are with the finite impulse response filters. So some signal processing algorithms also use infinite impulse response filters, which basically means that for when there is like a spike, it causes little oscillations, which basically means that if there whatever happens before the P wave can somehow affect the shape of the P wave. So the alternance that you see doesn't necessarily come from electrophysiology, that's there, but from something that's preceding it. So that's also something that can affect the analysis. And also, what I found quite particular about this method, that they focus like the way how it's designed, like that basically it's split out, like it splits the recording in even and odd beats. Then it's kind of biased, I would say, or it can be biased towards some repetitive beat-to-beat alternance. So if the alternance is, how to say, occurs throughout more beats, this is a behavior that the algorithm cannot really catch, right? Because it just kind of averages even beats and odd beats, and then it compares these two. So I don't know. It's just something when I saw the methods, I was wondering like whether this doesn't really little bit cause a bias in the method, or like bias in detecting certain types of alternance and attenuating other types of alternance.
So just to add a quick point in how the averaging is done, it's done in moving windows, so it doesn't split the entire recording into even and odd and then just average it. So based on the paper, it takes 10-second strips and takes 4 beats at a time, and analyzes moving windows of these 4 beats. So I'm guessing that helps sort of account for each sort of moving window. So each beat in general, and an alternance value is calculated for each beat. And secondly, the alternance value is calculated for each sample point in the P wave. So not just one point of the P wave, but all, say if it's a 50 millisecond window, then all 50 sample points of the P wave, and then an average is done. So it will take an average of the entire P wave also. So if there is some sort of alternation, it will capture it.
So let's assume, for the sake of the discussion, that this is technically correct, although Vladimir has pointed out various technical problems. Let's try to find a mechanism that can explain this. One thing that I thought of is that the heart rate may be different in the patients with and without arrhythmias, and that it is secondary to heart rate changes. Is that a possibility, or did they report a heart rate in the two groups?
So I don't think they've reported the average heart rates or RR intervals across the group, or mentioned any differences between the two groups.
Okay.
So sorry, Kanchan, just to add something to both the Ruben's question and Vladimir's comments, that was actually the thing that I was trying to say. Well, is there any, I mean, do they talk about any relation between QRS alternans and the P-wave alternans? I mean, on the opposite side, to check that if is the QRS alternans related to the P-wave alternans that it shows or not. First of all,
No,
and
so they don't do any, sorry to cut you off, they don't do any sort of correlative study. And in fact, actually, they just state that the pulmonary rate isolation reduced T-wave and QRS alternans, but don't really talk about or discuss that aspect of, was there something different in the patient population, some underlying pathologies that could have caused this? Yeah,
and to add something to that, actually, I think that the main reason that the number of the patient is not that much is because maybe the AI couldn't work that much good with so many patients. I mean, this is the thing, exactly the thing that Vladimir is saying. Maybe AI would be the only way to check the P waves because there are lots of errors and lots of factors that really, I mean, can really make it hard to understand what is the P wave and what is the difference. So, and I just wanted to ask another question. You talked about the second, I mean, there's another way of checking the alternance, and that was, as I understood, the Fourier way. I think, can you explain it a bit more than to check that if it is, I mean, can they do that to check the P waves or not? Because I'm not that much familiar with that.
Sure. So the spectral method is what like our group uses, and that it's what we used in our paroxysmal AF patients. So the difference, and I think why they use the modified moving average is, as I said, that this time domain method can be applied irrespective of heart rates. So it's a little curious to me now that they've not reported the heart rates in this patient population. So I'm assuming that it varied across subjects, and they couldn't precisely control it to the recommended range, and that's probably why they went with modified moving average. In the spectral method, essentially what you do is you create a sequence of say a window of 128 beats. If you're looking at the P waves, you take say, and you're just looking at the peak of the P wave, you take 128 consecutive P wave peaks, and you plot the power spectrum of those 128 data points. And in that power spectrum, you look for the 0.5 cycles per beat frequency. That's your alternance frequency. So you quantify the amount of power in this frequency, and the square root of that power gives you the alternance voltage. And there are like sort of other sort of variables. You sort of normalize it to the background noise. You can eliminate respiration and all of that. But in essence, it's plot the power spectrum of consecutive beats and quantify the power in the alternance frequency.
Okay, thank you. Actually, I'm not sure, but in this way, I think that there is no difference that much. I mean, if they can use it, they can use it to use the AI to use both of this way. Am I right?
They can. I think in the other paper, they mentioned why they went with AI and the modified moving average method. And it came down to this. It came down to not being able to control the heart rate across patients. And also that for the spectral method, you need a larger recording, sometimes a 2 to 5 minute recording, because you just need a larger sequence of beats. Versus here, I think they've looked at 10-second strips, which is, you can't apply the spectral method on that.
Thank you.
No problem.
For my first part was a little bit to the technical part. Now I started thinking a little bit more to the physiological mechanism, right? So the intervention there was pulmonary vein isolation, just pulmonary vein isolation and nothing more. No box, no other things, just pulmonary vein isolation.
Yes.
Right?
No, there are also boxes and additional lines.
Yes.
Some
I think so.
patients.
Okay.
In some patients, yeah.
In some patients.
Yeah.
Okay. So okay.
CTI also sometimes.
All 45 patients received pulmonary vein isolation, and then there are some subset of patients that received additional procedures for ablating the arrhythmia.
Because just thinking about it from physiological perspective, if this is the procedure or a procedure that reduces the alternance, so as Ruben mentioned, let's consider like the technical part is brilliant, and we just focus on what the signal tells us. Then it means that it's probably the pulmonary vein region which is responsible for the alternating part of the P wave, and this should be clearly visible on the P wave, because this is just intervention. If it's let's say dominantly the effect of pulmonary vein isolation, this should be kind of visible change in the, okay, maybe it's a bit more naive thing, but in the second half of the P wave, right? Because like whatever happens in the right atrium should stay the same, and it should only be the difference in the left atrium that we should see. So is there any information in the paper whether this kind of change in the P wave was somehow quantified, whether it's an overall change of the shape, or the change is just in the second part of the P wave, or how is it? How does it look? This change in P wave shape?
So they didn't quantify other P wave indices. They do talk about a little bit in the introduction that these sort of other P wave characteristics, its overall duration, this skew in the morphology. These were indices that were studied by other groups in other AF subjects, but I don't think anybody studied the effect of PVI, or pulmonary vein isolation, on the specific indices. So we don't have an answer to that.
Well, there were some examples. You showed some ECGs where it was clear, I think, that the last part of the P wave was involved, not the first. But that was only an example.
That was just graphic.
Maybe you can bring it up. But I think it's an interesting question, Vladimir. And I've been thinking about it. Would it be possible that there is an altered mechanical behavior of the atrium that secondarily leads to these P wave changes? A bit like what we studied with Lisa Gottlieb in Lyric. So by changing the stiffness of the left atrium around the pulmonary veins, that may be an antiarrhythmic procedure. And if you're not succeeding in achieving this stiffness, then maybe you fail the procedure. I'm just speculating, of course.
So there was
to
this
me,
sorry,
sorry.
go ahead.
No, to me, like when I just think about it kind of intuitively, if this is really the case, so the difference is, let's say, in the second part or second half of on the latter half of the P wave, then it's clearly the alternance, the source of the alternance seem to have cause in atrial repolarization, right? Because otherwise, because basically the first, like the depolarization, since it's a depolarization, since it's let's say P wave is mainly the depolarization, depicts depolarization, then what can change the shape of P wave is the previous repolarization in that region. So then to me it looks a little bit like if there is the alternance, and pulmonary wave isolation gets rid of the alternance, then to me it seems like it's an issue of repolarization.
I'm not sure, but because the left atrial area is also the last to be activated during a sinus beat, at least.
But on the examples that were shown, it was the tip of the P wave that was higher and lower. It was not the end of the P wave where the alternation was.
Okay.
I saw
Yes.
an example where there was a small P wave and a longer P wave alternatingly.
No, that was just a graphic I had given to demonstrate.
Oh, okay.
The paper actually, the alternance they show is just in the peak. That's where
Okay.
they find the max.
Okay. Yeah.
Could be the U wave of the T wave before.
Yeah.
As
But
there was
uh,
also there.
But sorry, as well, what I was saying actually, because I think that it is because of the repolarization. If we say that the atrial repolarization is inside the QRS somehow, then is it possible that the QRS alternance would be in real atrial alternance that is making QRS looks, I mean, maybe alternating?
Yeah, possible.
No, but also when you have, for instance, a long PQ time or a second degree AV block, you don't really see something after the P wave that could have such an effect on the QRS complex. So it's very low voltage.
Yeah, that's true. Yeah. I think we have to measure something to be able to assign a mechanism to this.
Right. Sorry, I'll just quote a study that was done to look at a similar correlation between T wave alternance and ventricular cellular alternance. There was a group that actually did sort of simultaneous mapping of sort of 128 points along the ventricle while they recorded ECG, and they were able to get a sort of more quantitative comparison of what happens when do you actually see T wave alternance, and how does it correlate to repolarization heterogeneities at the substrate level. So they were able to do a more sort of comparative link between that. So I don't know if that's feasible to do in atria because it's so complicated and the structure is so different, but sort of a simultaneous mapping at cellular and electrophysiological level would maybe answer some of that.
The problem in the atrium is that the unipolar electrogram doesn't give a clue about repolarization. That's really a disadvantage. Okay.
Yeah.
Sorry.
One final kind of remark, like point to the discussion. So this paper was published in the Journal of Cardiovascular Electrophysiology, right? Since it uses human data and I'd say quite a state-of-the-art signal and data processing techniques, including AI, I'm a little bit wondering, like, why this journal? Why not somewhere else? Because I can imagine that this is not a target journal for many researchers.
So I can answer that based on my own experiences, that because when you quantify something like P wave alternance and don't follow it up with some mechanistic insights, then a lot of cardiac journals are skeptical about just publishing that, oh, we found this phenomena, but how does that help us clinically? So maybe that's one of the reasons.
Okay.
Sorry, guys.
So
I don't know if you noticed, but I'm back. I
Yes.
arrived late.
well, welcome back.
I'm happy that the discussion seems quite on point. Did you spoke a lot about atrial repolarization, which was a topic that I really enjoy.
Yes, I think we try to address it, but it's very difficult to measure, of course.
Are you convinced in the end, Ruben, by this paper, or at least by some usefulness of the method?
I don't know, but I would really like to know the mechanism before I would work with the method.
Do you think that higher resolution P wave detection could be useful, like at higher frequencies that 250 cutoff?
Yeah, but then that's a technical issue. but I first would like to see the heart rates, that they are the same in the two groups, to exclude bias by that. And then I would prefer to do some mechanistic studies in animals, for instance, or maybe it can be done during PVI. I don't know. By pacing the right atrium and studying the P wave.
Okay, it's a good idea. We have these models of menopause, which are they have a quite large remodeling in both ventricle and atria. Could be useful to pace the different frequencies and see if there is like any kind of alternance. This we never thought about.
What kind of model was that?
It's a menopause. It's a ovariectomized animal.
Okay. In sheep.
A sheep, yes.
Okay. Yeah, that would be interesting. And I would also like to know whether, for instance, alternation in the left ventricular pressure wave was associated with the alternation in the atria. I mean, the heart it starts at heart rates of 120 beats per minute, I understand, and at that point there may be alternation in the mechanics, but not in the electrophysiology of the ventricles, and that might secondarily influence the P wave, I would say.
It's very interesting because we're gonna do PV loops in these animals.
Okay.
So it's definitely something that we can explore, and yeah, maybe we can talk about it later after the journal club.
Yeah, and then you have to pace from the right atrium at a high rate, so that you encroach on the ventricular repolarization in order to get an alternance.
Yes.
A ventricular alternance, and then see whether it occurs in the atria as well.
Right. Interesting. Yeah.
I don't see yet how that influences the success of PVI, but that's the next step.
Okay.
Okay. Any more comments from the clinical side, Guido or Chantal?
No, not really. From my side, I don't know. I'm skeptical about the duration of the recordings. I would really like to see something a little bit more heterogeneous passing this test, but maybe I'm just not in the field, and I'm too skeptical by nature.
I think we are all skeptical.
Okay, good.
Vladimir, by technical issues.
Yeah.
I'm skeptical because of mechanistic issues. Kanchan is skeptical all the way. So,
in total, we might make one optimistic person.
yes. Okay. Shall we leave it at this? If there are no more questions. Severine, are you listening in?
Yes, I'm here.
When is the next journal club?
The next journal club will be on the 24th of June.
Okay.
And the speaker will be Christelle Mimoun. And we will have the name of the article yet. Very soon.
Very good. Well, this was a very good journal club, I think, with a lively discussion. Thank you very much. And I hope to see you next month.
Thank you a lot. Thanks a lot.
Thanks.
Thank you.
Thank you.
Bye.
Thank you.
Bye-bye.
Bye.
Bye.